All right, good afternoon, everyone. We're going to be starting the session. So welcome to the Microbiome and Polymicrobial interaction session. Really happy to be there in person for the first time and also seeing all those familiar faces and faces I've only seen on Zoom. So for this afternoon, we have a couple of interesting talks that are going to be focusing on that microbiome and polymicrobial interaction theme. And I'm happy to have my co-chair. So Christina, I'm Fabrice. I'm a postdoc in Georgia Tools Lab at Dartmouth College. And I'll let Christina introduce herself.
Hi, I'm Christina. I'm an assistant professor at the University of Calgary and a clinician. I don't think we need to convince this group why this is an important session and why the microbiome and polymicrobial interactions are important. But from our perspective, what we really wanted to get out of these sessions, and I think we have quite a diverse group of talks, in particular around the microbiome, our evolving understanding and the importance of it in the post-modulator era and how we can translate a lot of this from bench to bedside.
Yeah, and the beautiful thing about the microbiome is that it unlocks the potential to really understand how interspecies interactions between microbes can shift a number of phenotypes that would not be readily seen using pure culture conditions. And with that, we're going to be starting. So for our first speaker, we have Karen Lester, that is a clinical project manager, that are going to be giving us a talk that is titled Striking a Ball NS, Combining Metagenomics.
And just a bit of housekeeping. So for the speakers, you'll have your little timer here, so we don't have to boo you if you're over time. And then if you have questions, please use the app, but otherwise we can go old school and you can just raise your hands, whatever you're comfortable with.
Okay, perfect. So I'm just using this to move through.
So this is the laser pointer?
Yeah.
So if you want to, you can either click on the laptop or you can just move with this.
Yep.
It's really up to you.
Yeah, perfect. Thank you very much to the organisers for having me here today. I have nothing to disclose in relation to this presentation. And I just want to let it be known that I am here presenting on behalf of one of my colleagues who couldn't travel. I am not a clinician and I am not a basic scientist. I'm a project manager, so I apologise in advance if I get something very wrong today. So I'm presenting on behalf of Jamie and his study, which is looking at combining metagenomics and culture methods in the paediatric CF airway. So the airway contains a series of connected but distinct microbiomes, and we know that these change as we move through the nose, the throat, and the lungs. The major taxa are well known because they are pathogenic or they are simply resident there, but there are also rare microbes that are not as very well understood. The pathogens or the microbes that are there are likely commensal and protective, and we know that the microbial diversity develops with age and peaks around adolescence. And some studies have shown that the respiratory microbiome can also change when disease is introduced into the environment. So surveillance of these microbiomes aids management of pathogen burden, treatment, and follow-up. So in the context of CF clinical care, microbial culture surveillance is a routine practice. We do this by taking different types of samples, be it expectorant sputum, nasal swabs, throat swabs, or bronchoalveolar lavage. And we examine these samples using either microbiology techniques or molecular biology techniques. In the paediatric setting, a BAL can be very onerous, can be complex, and we wanted to see if there was a surrogate that we could use to look for these microbes in the paediatric setting and how well do these alternatives compare against BAL. So this study is part of SAINTS. So SAINTS is a study of airway infection of the nose, throat, and sinuses. And the objective of this study was to profile the respiratory microbiome in this paediatric cohort. We also then wanted to see how closely we can match metagenomic culture surveillance data with microbiology culture surveillance, and then how well do throat swabs and nasal swabs perform alongside BAL samples. So for this study, there were 30 participants, so a relatively small cohort, very preliminary data that we got out of this, and we collected throat, nose, and BAL samples concurrently at one visit. So the sampling, as I've just said, samples were collected during a routine bronch examination, which were done as routine clinical care in children under the age of 6, and the children were not undergoing any exacerbation at the time of sampling. The 3 different types of samples were collected, and the DNA was extracted using 2 different methods or 2 different kits, so the complete 5 hemolysis kit and the master pure DNA isolation kit. The DNA samples were then sent to our collaborators, and the sequencing was performed using Illumina NextSeq platforming. And when we removed host reads and performed QC, we had some loss, but we ended up with 30 billion reads post QC and host removal, and we aligned these then with the Kaiju reference database to identify the data sets that were there. Abundances thresholds were then based on total community relative abundance. Microbiology culture surveillance was then performed on site in the Children's Health Ireland Hospital Group, and they used selective, semi-selective, general media, and specific PCR for known microbes to identify these microbes in these environments. So our first question, or first thing we wanted to look at was the composition of the microbiomes between bowel, nasal swab and throat samples. So what we observed was that the major taxa were consistent across the lungs in all of the children in this cohort. However, what we did then see was that the throat swab had a clearly more diverse microbiome when compared to the bowel and nasal swabs. The composition of bowel and nasal swabs were then notably more similar to each other than to the throat swab, but there was no stratification of alpha diversity when we compared age of the participants in the group. So in figure A1 here is just kind of a visualization of what I've just said. So we see that the alpha diversity of the throat swabs, which is in green, is visually higher than that of the bowel and nasal swabs, which are more closely correlated in this instance. We then also looked at, in figure B1, we looked at the relative abundance of the top 14 bacterial in these environments. And what we've seen was that across each of the sampling, there are comparative. We also can see that in individual samples within the different sampling groups, they were also comparative. However, it's quite clear then, if we just look at this, that our throat swab sampling is still slightly variable when compared to our bowel and nasal swab. So then we wanted to align microbiology culture surveillance and metagenomic culture surveillance. So we took the top pathogens that were surveyed in the CF lab and looked at whether these were also identified using metagenomic data. So this is what we're looking at in figure A here. And what we saw was that for all of the bacteria that were identified using microbiology culture surveillance, they were also identified using metagenomic culture surveillance, but the converse was not true. So the stars in this plot denote where microbiology culture surveillance identified these bacteria. So it's there are instances then where bacteria were identified using genetic sequencing that weren't actually picked up when we did just microbiology culture surveillance. With that in mind, then we wanted to look at what are the abundance thresholds that were necessary to then detect the particular bacteria in both culture and metagenomics surveillance. So what we observed was that the thresholds had to be significantly higher in those for specific bacteria, for the different bacterias, in order for them to be detected across both of the surveillance techniques. And it obviously differed depending on which bacteria you were looking at. So then our next question was, how comparable are the sampling methods? So how do throat swabs, nasal swabs and bowel compare against each other when we want to identify microbes in these areas? So what we noticed was that the microbiome was very similar between the nasal swabs and the bowel samples, with a large degree of overlap. But we also noticed that all three communities, so be it from the nasal swabs, the throat swabs or the bowel samples, were still significantly distinct from each other. The nasal swab provides a strong proxy for the lung state, and nasal swab and bowel sampling gave similar correspondence for reference metagenomic sequencing data. So in this NMDS plot here, what we can see is that the strong similarity is shown between, on the left-hand side, we can see the strong similarity is shown between bowel and nasal swab samples, whereas we see less similarity in terms of our throat swab samples. They're more variable and more diverse. And then in B, we were looking at the comparison of these different sampling types when looked at their bowel culture surveillance. And we see that bowel and nasal swabs are more closely related in terms of their similarities and what we see again. And throat swabs were slightly less similar to bowel and nasal sampling. So what we determined from this preliminary data was that using metagenomic sequencing, bowel and nasal swabs were more closely related in the pediatric CF cohort that we were looking at, while the throat swabs were much more variable. But nevertheless, the respiratory microbiomes were still distinct, significantly distinct on their own as well. So there were several classic pathogens that were shown to be commensal-like, but we did look at them at a genus level and not at a strain level, which was obviously a limitation to this study. When we wanted to align the culture and sequencing surveillance, there was no single level for monitoring that allowed us to identify all of the bacteria that were present in each of the environments. Some of the clinically relevant taxa were not, so some of the clinically relevant meaning the taxa that we look at in culture care as standard care were not always detected using culture surveillance, but were detected using genetic sequencing. BAL and nasal swab sequencing data match culture surveillance much better. BAL and nasal swab sequencing data match culture surveillance of BAL equally as well. Obviously one of the questions that we wanted to ask was how would surrogates for BAL compare in their ability to identify microbes in these areas, and it might be a consideration that we look more closely at nasal swabs over throat swabs in certain settings where BAL might be difficult or not feasible. There are obviously limitations to this study. The limited study size, the N is quite small, it's just 30 patients. The loss of sequencing data when we were QCing, and then we focused on identity of these microbes over their function. We also didn't have a control reference group at the time of sampling. So I just want to thank our collaborators and our PI and our funders for this, and you for listening. Thanks.
And now we have a few questions on the chat. So first question from Laura Brenner. So the question is, any attempts to look at the absolute abundance, or was abundances only done with relative abundances? Were your reads depth different between different types of samples? So basically, I think she wants to ask. Oh, sure. Is this working? Okay. So any attempts to look at absolute abundance, or was abundances only done with relative abundances? Were your reads depth different between different types of samples? Did you get more reads in the throat swab versus the nose swab?
The read depth was the same across the samples. I can't comment on the total abundance. I didn't actually do that analysis. I can find that out for you. I apologize. I'm not entirely sure on that.
Any other questions?
I have a question.
Mm-hmm.
I'm just curious, for your throat swabs, were these like oral pharyngeal swabs or cough swabs, or were they consistent throughout? Because I'm wondering about some of the variability there.
So they were consistent. They were all performed at the same time as the BAL and nasal swabs. So the patient was already in, so they were in cough swabs. They were oral pharyngeal swabs, and consistent across the. The only limitation might have been the nasal swabs, which is the size that we were going for mediation. But just with terms of like patient age, etc., just access for that.
I also have a question. So I was wondering, are there like any microbial taxa that were overrepresented in your different sample types?
Overrepresented?
Yeah.
In terms of in the specific environments, were there one that was in the culture or the metagenomics data?
More with the metagenomics data?
I'm not sure, so I'm not sure. Sorry.
Questions? And could you just like speculate a little bit about which microbes that were detected using metagenomics and not like how cultures were? Like, do you have any sense? Jesus, I'm not used to speaking in a microphone, sorry all. Do you have any sense or idea about the microbes that were detected using metagenomics that were not detected by culture methods? Which are those microbes? Do you have any idea about those?
So some of them that were shown there, so E. coli was only detected in one sample across BAL, nasal and throat, but it was actually detected across all samples using metagenomics, so that was, it was less abundant, so the abundance wasn't high enough to detect it in culture. And it was the same we saw the same in strep and staph as well in samples.
We don't have any other additional questions in the chat. If people want to ask a question and raise their hands too, that's completely fine. Other than that, we're just going to thank our speaker. Please, let's applaud.
Thank you.
So our next speaker is Dr. Sophie Darsh, who's going to be telling us all about Pseudomonas and Aspergillus interactions.
Good job.
There we go. I'm the first one to do that at this conference. There we go. It is 15 minutes. Good. Okay. Is this working?
Yeah, it's a laser pointer.
Okay. There you go. Okay. Okay, so little welcome there by falling onto the stage. That's what happens when you do too many virtual conferences and you're stuck behind a desk. You forget how to walk up and greet people. My name is Sophie Darch. I'm an assistant professor at the University of South Florida in Tampa. My lab's still relatively new. We opened about 3.5 years ago now. So what I actually wanted to do today was kind of share with you some of the tools that we're using in our lab to really start to study these microbial infections, and also kind of touch on some new data that we have from one of my new graduate students. It's not working. Okay, I have no disclosures. Okay, so I'm going to jump right on the ecology train and talk about bacteria in nature. So we're obviously at a CF conference, but if we think about bacteria in nature and how they might exist in a host normally, I think it's really important to think about how they're actually organized and how they grow. And so there's some really great examples now across different host sites of how bacteria will organize themselves and ultimately how this impacts their interactions with each other, virulence and pathogenesis. So this is an example of the oral cavity. You can see here 2 different streptococci species, and you can see how they organize themselves in this circular pattern, which really ultimately impacts the virulence of one compared to the other. Another example is the gastrointestinal tract. Here you can see every color represents a different taxa. And the important thing to take away from this image is that you can see how the different species basically co-organize or organize themselves into these different groups next to each other. And ultimately, again, there's results and differences in virulence and pathogenesis. This isn't just localized to thinking about human pathogens. Sea kelp, which again is not related particularly to the CF lung, but it is a good example of showing you that these types of spatial distributions are not limited to human hosts. And so this is something that we can really look at in a lot of different settings, but again, you see this localization or spatial structure. The main thing is with this spatial structure, even though lots of organisms adopt this, we still don't understand the mechanisms that really underlie this. And this is really important for thinking about bacterial interactions. That's what we're talking about in this session today. And when I talk about bacterial interactions, I mean interactions that can happen between the same species, a bacterial species or microbial species with the human host, and then those with other microbes. And these interactions are all mediated by this idea of spatial structure. And that can be how the bacteria actually grows, so thinking about its physical shape, size, how it's attached to each other in a small community. We talk about biofilms a little bit in the next few slides. But also how the actual tissue that an infection is in or residing in can also influence the shape and spatial structure of those interactions. So in my lab, we study Pseudomonas aeruginosa. And what does that look like during actual infection? Well, if we look at infection in the CF lung, we find now that Pseudomonas grows as these small, dense aggregates of around 10 to 1,000 cells. So on the left, I have an example of a piece of explanted CF lung tissue. In red is Pseudomonas aeruginosa, and you can see it forms these small microcolonies in blue are stained host immune cells. And then in CF sputum, if you take that directly, look at it under a confocal microscope, you can see again in red, these aggregates, these can be free-floating or attached, again stained are host immune cells. So you can see this aggregate phenotype in these 2 direct settings. So in the lab, we use a synthetic CF sputum media. Some of you might be familiar with that. There are a few different types of it around in the research world. Ours was developed by the Whiteley lab, where I did my postdoc. And basically, they took sputum samples from CF patients. They did a chemical analysis. And then what they did from that was create a defined media. So it contains amino acids, salts, mucin, DNA, and lipids, which give it its viscosity. It is a defined media, so from a research perspective, it's really great. We can take things out, we can add things in, and know what the ultimate consequence of that is. And now there's been a lot of work looking at Pseudomonas in terms of how it responds and how it grows in this synthetic environment compared to actual sputum. So I spent some of my postdoc showing that aggregates appear to be the same size in our sputum media at certain time points or a range of time points compared to actual CF
sputum. And then also from the transcriptomics side, Pseudomonas responds very similarly in our synthetic environment compared to actual CF sputum. So in the lab, we basically take these chamber slides, we have our synthetic sputum media, and then we inoculate with Pseudomonas aeruginosa. And then we use confocal microscopy to really study these aggregates. And again, you can see, this is after about 4 hours incubation. Looking at confocal microscopy, you can see these aggregate structures which have grown, and you can see a range of different sizes, again, free-floating or suspended in this viscous media. And so what we like to do is then take these images and then look at them at the individual aggregate level. So with image analysis, you can really start to ask about community or aggregate functions, aggregate phenotypes, thinking about the volume, shape, and position of these different aggregates that we can culture in synthetic sputum. And so we can grow these aggregates. What can we do with them? Well, we can study things like antibiotic tolerance, host immune cell tolerance, or we can study things like microbial interactions. The nice thing about using this system in terms of spatial and temporal resolution, in one experiment, we can analyze around 50,000 aggregates. So it definitely gives you an idea of how this community is behaving as a whole in terms of thinking about functional outcomes, but also how those individual aggregates are behaving by themselves. So moving into the Aspergillus part, we're not a fungal lab, I will say that outright. We're a Pseudomonas lab. But when you have a student with a great idea and you're starting a lab, you think, hey, let's branch away a little bit from what I did during my postdoc. But spores go everywhere, I will tell you that now. So we are working with Aspergillus fumigatus, which is unfortunately one of the more easier ones to work with, and it is an emerging pathogen in CF. So for those who don't know too much about it, it's a filamentous saprophytic fungus. It is part of the normal flora in humans, so existing in the mucous membranes of our upper respiratory tracts. We breathe in so many spores every day in a healthy airway. But ultimately, the conidia, which are those nice kind of flowery-looking parts at the top, enter the lungs. They germinate on the alveoli, and then produce mycelium, which are the long, stringy bits which penetrate into the lung tissue. Now, for those physicians in the audience, you'll all be familiar with this bronchopulmonary aspergillosis. So most common in those whose immune systems are compromised. And so this is the new problem in our subset of CF patients or individuals with CF. Really, some people are discussing this as a result of these new, more effective modulated therapies opening up a niche for these emerging pathogens. So according to CF Foundation's website, it affects around 2 to 19% of the people with CF, and it is more common in those over 6 years. So those are kind of the statistics in terms of thinking about this from the CF perspective and why it's important. Thinking about why it's important in our lab, and we're really interested in studying these microbial interactions, if we think about this in terms of what we know about Pseudomonas aggregate formation, we know that individual cells form aggregates. These mature and disperse and go on to form new aggregates in the context of a space within an alveolus. This is a little bit of a dramatization for this picture on here, but it gives you the idea of what these potential interactions then of Aspergillus with Pseudomonas may be. So this idea that Pseudomonas could kill Aspergillus on direct contact. You see that localization down there on the bottom right. The idea that Pseudomonas and Aspergillus could actually coexist. So living on or socializing with the hyphae without actually killing the AF. And then the idea, we know that Pseudomonas produces a lot of very nasty virulence factors, so producing volatile compounds that could actually either promote Aspergillus growth or decrease it. So these are the things that we don't really know and kind of what we're looking to answer question-wise. So we're studying Pseudomonas and Aspergillus in 2 ways in the lab. First, we put it in our synthetic sputum media, and we saw these first images. So in green is Pseudomonas. You can see this kind of little overlay with the Aspergillus hyphae, and you can see that it looks from like top down looking some localization. We have another project looking at lysogens and trans-kingdom interactions. That's for another day. What we're really trying to find out now, and I'm going to touch on now in the next few slides, is thinking about co-infection with Pseudomonas and Aspergillus in this sputum media overlay model. So this is kind of a schematic of our overall goals of the project. So really thinking about not just incorporating now Aspergillus into our synthetic sputum media, but also host cells as well. So really get that idea of a host response and the impact on the Pseudomonas aggregates and the Aspergillus co-infection. So we've done a lot of confocal laser scanning microscopy in the lab. We do a lot of imaging. But what we're doing with this project is actually taking the supernatants from the cell fractions and from the supernatants and looking at it with proteomics. And then ultimately the endpoint of this will be to challenge these cultures with antibiotics and look at the surviving biomass. Today I'm just going to focus on this confocal part just to give you an idea of how we can observe these multi-kingdom communities and as a tool for really resolving these interactions. So time-lapse imaging, this is something we use a lot in the lab. This really just gives you an idea of one of the cultures we had of Aspergillus and Pseudomonas. In green, you'll see these hyphae. Also green, I'm thinking are some conidial spores. In the pinky red, I'm sorry for anyone who's colorblind. Once I made these slides, I was like, this doesn't make any sense to do this again. But pink, red is Pseudomonas aggregate. So what you can actually see is some sort of localization or co-localization with those hyphae. This is a little bit more of a closer view. Again, you can see this. So this is taking images probably every 25 minutes, I think, from what I remember. But you can see some aggregates getting a little bit bigger. You are seeing the hyphae stay around the same size, but you are seeing this, not necessarily, I won't say any sort of adhesion or direct contact with the hyphae, but you are seeing some co-localization. Again, this right now is very qualitative looking at these images. What we can do is start to quantify things like this. So really what we're thinking about, like I said, I'm not a fungal biologist, but looking at it from the fungal perspective, looking at metabolic activity of fungi during this co-infection. So again, if you separate out the hyphae of the Aspergillus, we can use certain stains and use intensity of that stain as a proxy for metabolic activity. So you can see here a range of colors. Really what this is showing, the red is really showing you the highest activity along those hyphae, where Pseudomonas has actually been in culture. So this isn't a monoculture. I've just taken the Pseudomonas out for now for looking at it. But you can see the red is probably the activity is the highest compared to other parts of the hyphae. So this is an interesting perspective to get some actual quantitative data of these interactions that we now have in the lab. So as I said, intensity of this FUN1 stain can be a marker for metabolism and used to observe this metabolic activity in the hyphae. Again, we can look from the Pseudomonas perspective, which is what we're doing primarily. So the hyphae here in green, the aggregates are in white. I realized after I put this talk together that I color-coded the wrong part. So you can imagine that the aggregates can actually be color-coded by volume, by different, if you have a gene reporter, you can color-code all these different things and really look at it from the perspective of the aggregates themselves, the individuals, and get that quantitative data out. So this can identify co-localization and potential interactions and really start to kind of bridge that gap between looking at spatial organization and what is the actual functional outcome of individual aggregates, or if you put that aggregate population together. So this is one little bit of quantitative data that we do have so far. On the y-axis is aggregate volume. Across the x-axis in time. So this is taking some of these images as representative for this data. And you can see over time what this is showing you that the Pseudomonas aggregates are getting a little bit smaller during their co-infection with Aspergillus. They are there, they are coexisting, but they are getting a little bit smaller. So of course, thinking about this, there's a couple of things that came to mind when looking at this data initially. Potential interactions, so Aspergillus produces gliotoxin. That's definitely something that we'll be looking into in the future to think about what might be mediating these interactions. And then from our previous work, the maximum size we see here of around 60 microns or 60 cells is actually a bit smaller than what we've actually previously seen in our synthetic sputum of Pseudomonas alone. So it is interesting. There seems to be some impact, and at least that we can observe and study this further. So we've just started integrating our human respiratory epithelial cells. This is just to remind you of that snapshot of sliced CF lung tissue. And now we have respiratory epithelial cells. You can see the nucleus stained in blue and our aggregates surrounding. We're using primary small airway epithelial cells for our control and CUFI bronchial epithelium cells for our CF line. Ultimately, what we've done is adjusted our synthetic sputum media to make the host cells very happy and survive and not come off the bottom of the tissue plate, which apparently is not a good thing when that happens. And so my grad students have been working on this to really start looking at Pseudomonas and Aspergillus in this setting. And this is just my very last kind of preliminary data slide of really how we're using this as a tool to look from the proteomics perspective. So are there potential protein interactions that we can look at now for the mechanistic underside of these interactions? So this is just from PA monoculture, where we normally just grow it in SCFM2 without those host cells. Adding in the host cells, now we can actually detect certain factors or proteins in abundance that are just because they're with the host cells and the actual synthetic sputum too. And some interesting things have come up so far to do with quorum sensing, with stress proteins and dispersal motility. So these are kind of the areas that we're focusing on. But I really just wanted to put this in as an example of the kind of data that we can get out of these actual in vitro setups. So in summary, it's flashing at me. Okay, whew. Okay, so Pseudomonas aeruginosa exists as aggregates in the CF lung. That's what we've been studying. Well, I've been studying since my postdoc and my PhD work. So now moving into my own lab, we're starting to think about emerging pathogens in CF and studying these microbial interactions. There is a need for systems and tools to understand aggregate interactions and really a higher resolution than what we have available now. Obviously, there's high resolution techniques thinking about genomics and metabolomics, but really thinking about how those bacteria, how they sit next to each other, how they interact, I think is really important from an ecology perspective that can also be applied to translational projects that really talk about human health. And so we're using high resolution microscopy and proteomics to answer many of these questions. I touched on this newly adapted version of the synthetic sputum we're using to work with lung cells. And our preliminary data indicates co-infection that we can study with this reduction in Pseudomonas aggregate size over time. So I'm confident that we're going to be able to unravel some of these interactions by using this system. And again, using proteomics, we've found a couple of target proteins in high abundance that really kind of point to some sort of interaction by using this environment specifically to mimic a little bit more closely the CF lung environment than using traditional lab cultures. I mean, one of many goals of a new lab, understanding mechanisms of these interactions, and really it will help us understand persistence and potential new therapeutic targets. So with that, acknowledgements, my lab, who are not, they're real people in real life, they're not just cartoons. University of South Florida has been very wonderful in getting my lab started up. Maggie is our honorary member. She took part in this Great Strides Walk a couple years ago. And of course, to my funding sources, the CF Foundation has been wonderful in supporting us getting our lab up and running, and we really do appreciate that. And thank you very much for your attention.
That was an excellent talk. Thank you. And you were very popular virtually. So
Ooh.
Many questions are rolling in.
That
so
makes a
we'll
change.
go through. So first question is, are there impacts of co-culture on the abundance of the two species? Meaning, do the total number of bacteria or fungi differ, implying there's competition?
Ooh. So we have done some, I mean, with the imaging, we can do, total biomass of both. I haven't quite worked out, I could do Aspergillus by total volume, so I haven't looked at it from the Aspergillus perspective. I think it depends also on how you set the experiment up. So right now, we're adding the Aspergillus in first and then adding Pseudomonas afterwards, just because of the growth differences or the growth rate differences. But once Pseudomonas is in there, it seems to get to log phase and then stabilize. So it's certainly not being killed off. But we haven't done those direct competition experiments yet, but we definitely need to do those. That's a huge part of it.
All right. Excellent. So next question is, do you see an oxygen gradient develop in your synthetic media? And if so, have you seen oxygen affect these pseudomonal Aspergillus interactions?
So oxygen gradients is something that we've always thought about kind of mimicking, obviously thinking about the infection environment of the CF lung. We know that an oxygen gradient does not exist or start to exist in the sputum media until about 12 to 14 hours. So all of our experiments we're doing before that time point, mostly, if we're doing things like the previous work we've done on quorum sensing and antibiotic tolerance have been those shorter time periods. I imagine it could potentially have an impact. I don't know the answer to that question because we haven't tried it, but it would make sense that it would. I think using something like that metabolic marker that I showed you on the Aspergillus will be a great tool to look at that in terms of the Aspergillus, how it's interacting in those spots, and then also we have some great oxygen reporters for the Pseudomonas that we could definitely look at that. I know the answer to the question because we had a reviewer for a paper who asked that question, so that's why I know that one.
Great minds think alike.
Yeah.
All right, next question. Although the molecular interactions show Pseudomonas aggregates getting smaller in synthetic sputum, We see that Pseudomonas and Aspergillus co-colonization is very common clinically and may be associated with worse clinical outcomes. Can you comment how these interactions in vitro may translate to humans, or maybe what you postulate might happen?
Yeah, I mean, I think that there's definitely some things that come to mind first with that question. It's a great question. Thinking about, so if you have this idea of hyphal, Pseudomonas aggregate interactions, the aggregates are smaller. Most of us can think about small colony variants, then the clinical correlations to that. Aggregates themselves have been, growth as an aggregate has been correlated to increased antibiotic tolerance and host immune cell tolerance. So it could have some role in terms of thinking about persistence of infection. Obviously, when we're thinking about the clinical stage of organisms that are there, Pseudomonas has been there first, Aspergillus comes in, there hasn't been an obvious displacement, so Pseudomonas is still pretty tolerant to a lot of things. So I couldn't give a direct correlation, though, between antibiotic size and tolerance, although we do have some quite nice preliminary data that does maybe indicate that. So
excellent. Okay. Next question. Have you tried adding characteristic molecular patterns of fungi like chitin? Chitin? Would the Pseudomonas behavior be similar?
We haven't tried that. That would be really cool to try, thinking about some sort of scaffold. We did have an idea of doing something like that with probably way more fancier than it needed to be with like hydrogels and things, but chitin could be pretty cool to try. Yeah, I mean, that would be, something, making some sort of in vitro matrix and then looking to see attractants. We would be able to track that. We've shown previously Pseudomonas being attracted to, human neutrophils being attracted to Pseudomonas. So we can look at those sort of tracking phenotypes or movement phenotypes in our model. So we could definitely try that. That would be interesting.
Your next experiments.
Yes.
Next question, that's quite popular. Have you binned FUN1 high and low populations and quantitated localized bacterial adherence / aggregation to describe the relationship?
No, we haven't done that, but that is also a very, I should get like a grant proposal notebook here or something. I think that'd be great. I was going to say, no, I mean, we could definitely do that. So again, it's hard to kind of show an audience the different quantitative tools that you could have, but definitely by using any kind of marker. So whether that was, I showed you a FUN1 stain, which is used in the fungals. You could use, expression, like LASP expression, to look at quorum sensing in one part of an aggregate compared to another, and then you can pinpoint that area and then look at everything else around it. So the beauty of using things like that as quantitative tools means you have that data set that you can use in many, many ways. If you have 50,000 aggregates in an experiment, your N's pretty high. So again, statistically, you don't need too many replicates to say you're quite confident with that data, I think, too. So that's also a nice aspect to it. But yeah, we could definitely, we could do that with the data that we have, just something that we haven't done to visually show you today.
Sure. And maybe last question then, and then you're out of the hot seat. Do the mechanical forces in the airway lung alter aggregate size or formation, and then subsequently the interactions between the two species?
So I don't have any data on this from our lab. Thinking about it, though, just thinking about the question, I mean, by mechanical forces of changing sputum, we still don't understand a lot about aggregates in terms of how, we know that they disperse, but we don't really know how they disperse, if it's a active process of breaking a chunk off that aggregate or single cells coming out. So we don't know that process. But thinking about actual movement, it could disrupt the size of the aggregate, either releasing a dispersed cell to then go and relocalize with another hyphae or conidiaspore or something like that, or making those aggregates smaller and then going, that kind of gives a nice round robin to that question on, maybe increasing antibiotic tolerance if you're a little bit smaller. So very active, dynamic environment, and I'm sure all of these factors play a part. So we did actually talk to someone at a poster earlier about rheology, so maybe that's something that we can collaborate on if she's in here.
Amazing. Well, thank you so much, and please thank our speaker.
All right, so now our next speaker is going to be Dr. Don Sanders from the Indiana University School of Medicine. And while I'm loading this file, the title of his talk is Effect of Therapeutic Antibiotic Exposure on the Oropharyngeal, that's a mouthful, and fecal microbiota in infants with cystic fibrosis. So.
Okay, all right, thank you. Well, thank you for the opportunity to speak today. I'm going to present on behalf of my collaborators, almost all of whom are from the University of Washington in the Hoffman Lab and the Microbial Core with Steven Salaponte. So I don't have any relevant disclosures for this work. I talk a lot about antibiotics in children with CF, and we use a lot of antibiotics in children with CF. So that's going to be one of the focuses for today. So as of right now, they play a key role in exacerbation treatment, and we're frequently using them in children with CF and especially infants with new onset symptoms. And historically, even children with negative cultures have improved with antibiotics. We typically use antibiotics to target pathogens that we've detected by culture with all the limitations that go on there. Most commonly, especially in young children, are staph and Haemophilus. And the most common class of antibiotics used are beta-lactams. However, there have been multiple reports and well-known information on unintended effects of antibiotics, including selection for resistant bacteria, organ damage. We think about kidney injury and liver injury, as well as antimicrobial effects on other microbiota that are not the intended target. Maria Nelson in Luke Hoffman's lab published this, or led this analysis of sputum microbiome changes with inhaled tobramycin. And so I'll show several of these plots today, these PCOA plots. So each different color represents a different stage in the treatment of inhaled tobramycin. So before week 1, 2, 3, and 4, and then follow-up. The large circles represent the average of the each individual, and then the small circles represent each individual sample, an individual person. So in this figure from this 2020 paper, she showed that the samples obtained off tobramycin and clustered together, whereas the samples on tobramycin clustered together, and these were significantly different. They also noticed that the largest changes were in untargeted oral and nasal species and not necessarily on Pseudomonas, which is what we think we're using the inhaled tobramycin for. And thinking about the CF fecal microbiome, the Hoffman Lab and others in this room have identified fecal dysbiosis in children with CF. This figure on the right shows different relative abundance from infants with CF and control infants. You can see the much higher relative abundance of the dark blue proteobacteria in infants with CF. This dysbiosis correlates with worse inflammation and growth, and in infants it's seen even in those who have never received antibiotics, so more directly related to CFTR and those effects. However, on the other hand, studies of the fecal microbiome in adults have definitely demonstrated the contribution of antibiotics to dysbiosis. So in a cohort of infants with CF, we wanted to address these questions. How does the typical beta-lactam treatment we use for respiratory symptoms in infants and children with CF affect their oropharyngeal swab microbiome, and I might say OP to avoid the mouthful, their fecal microbiome, as well as the complement of antibiotic resistance genes in each space? So the study design for this analysis, we had 5 infant controls who were enrolled after newborn screening and followed for a year, and we obtained samples at multiple time points, and these had never received antibiotics. So they weren't randomized to this, they just, in that year, hadn't received any antibiotics. And then we had 18 cases who had beta-lactam at least once in that first year. So we had serial collections of oropharyngeal swabs and fecal samples, and for the cases, we had typically 1 to 2 or more samples obtained while on antibiotics. And then we were also, among those cases, able to obtain samples off antibiotics as well. So we had 74 oropharyngeal swabs from 18 infants with CF, 14 of whom received beta-lactams for an exacerbation, and there were 44 swabs from those 14 infants, and then there were 4 infants who never received antibiotics, and we were able to collect 30 swabs from them. We had 74 fecal samples from 14 infants with CF, 9 of whom received antibiotics, and we had 45 samples from those 9 participants, and then we had 5 who never received antibiotics, and we had 29 samples from them. We then tried to compare we tried to make several comparisons, So we had the control group, which never received antibiotics. We had samples that we obtained on antibiotics, which were collected during treatment, and then we had samples that were off antibiotics. These were collected after antibiotic treatment. And so we were able to look at a couple of comparisons. So when we look at the samples obtained on antibiotics versus the controls, we can think about the short, very acute effects of beta-lactam treatment. And then we can compare the samples obtained off antibiotics or after antibiotics versus the controls and think about the residual, more long-term effects of beta-lactam treatment. For this study, Maria Nelson and Hilary Hayden from the Hoffman Lab did the majority of the work for us. So the DNA was extracted from samples, and then they used a method to minimize DNA from dead cells from the oropharyngeal samples, and that was really a key to be able to do this project. We looked at total bacterial load, 16S RNA, as well as we did shotgun metagenomic sequencing, phylogenetic analysis, and then we looked at encoded antibiotic resistance genes. So first, for the total bacterial loads, for the oropharyngeal samples, we have the three groups here, the controls in purple, the off antibiotic, and then the on antibiotics in orange. And compared to controls, there was a significant decrease in total bacterial load only among those samples obtained on antibiotics. On the other hand, when we looked at the fecal samples, the total bacterial load was decreased both among those samples obtained on antibiotics and those obtained after antibiotics. If we again look at the PCOA plat for the oropharyngeal swabs, we can see that the controls clustering over to the left in purple, the on antibiotics way over to the right, quite a distance, and then the off antibiotics sort of more in the middle between the two. So a couple of notes. We didn't have any reads for Pseudomonas, and that corresponded to throat swab data that we had. We didn't have any Pseudomonas in these infants. Only one child had Staph aureus detected, and that was very low relative abundance. 13 of the 22 or so had Haemophilus relative abundance more than 1%. And these were the major taxonomic drivers that were associated with these differences between samples. And in all cases, these were decreased in the samples that were on antibiotics. If we look at then turn to the fecal samples, we again see the controls now here are more to the right, the on antibiotics in gold off to the left, and the off antibiotics is more, I would say, to our interpretation, seems more closely aligned with the on antibiotics than we saw in the oropharyngeal swabs. In this case, there were taxonomic drivers that were both increased as well as decreased in relative abundance in those samples obtained on antibiotics. And then I'll turn to the antibiotic resistance. So we used this comprehensive antibiotic resistance gene database and mapped our sequencing reads to it. We normalized the data to single copy genes with this music, and then genes with the same card type were combined. So if there was a TEM resistance gene 1, 2, 3, and 4, and 5, that was just collapsed down to the TEM. We had about 500 gene types included in analysis from both sample types. And then we looked at gene abundance by those antibiotic status groups we've been talking about. So we have a microbiota dissimilarity index, and then we looked at differences in individual taxa with Wilcoxon rank sum test. So again, these PCOA plots look pretty similar to the microbiota plots. In the oropharyngeal samples, the controls and off antibiotics are very closely related, whereas the on antibiotic samples are much further away. In the fecal samples on the right, there's a difference between the controls and the on antibiotics, but the off antibiotic samples are more closely associated with, a little bit more separated from the controls. And to our interpretation, these patterns of changes in the resistance genes reflected those of the microbiota that reflected the patterns of change in the microbiota. We came up with about 10 significantly different antibiotic resistance genes in both the oropharyngeal samples and the fecal samples. I think boxed in red are those antibiotic resistance genes that target beta-lactams. So those are really the minority of the significantly different antibiotic resistance genes. So the differences on the previous slide were not driven by selecting taxa carrying the beta-lactam resistant genes, but actually altered abundance of encoded resistance to other antibiotic classes. So the summary of our findings, our results suggest that the beta-lactam antibiotics changed oropharyngeal microbiota and total bacterial load during treatment, but there was at least a partial rebound after antibiotics. Similarly, beta-lactam antibiotics changed fecal microbiota and total bacterial loads during treatment, but both seemed to rebound less after antibiotics than in the oropharyngeal swabs. So our conclusion is the effect of beta-lactams on fecal samples may be more durable than in the oropharynx. We do have some opportunities to investigate this further. The long-term effects of antibiotics on the GI tract may be able to be explored through the PROMISE and BEGIN studies. And then we'll be able to look at changes in the oropharyngeal microbiota with antibiotics in an upcoming already funded study with the title Oropharyngeal Metagenomics of Pulmonary Exacerbations in Children with CF. This would be a little bit older age group. So I'd just like to say thank you for all the collaborators on this project, including Cindy Ross, our research coordinator, our lab manager, and Misty Thompson, a research coordinator at IU. We had participants enrolled at other sites, including Nationwide Children, Cincinnati Children's, and University of Wisconsin, and then the Hoffman Laboratory that did really the majority of the work here, and the UW Microbial Genomics Core, and then all the studying participants and families. And I'm happy to take some questions.
Thank you for that fantastic talk. So one question that we have is, what factors determine the of antibiotics group? How much time elapsed after a course of antibiotics to qualify?
So how much time could pass after the completed antibiotics to be on? I mean, it could be as little as 1 day, but I think in reality it wasn't that short. And I think there was a pretty wide range. On average, it was about 30 days following completion of antibiotics. And it just, we see these infants very frequently in clinic, and we are sort of at the mercy of when the antibiotics occurred clinically.
Have you also collected any samples to look at inflammatory markers and see if there are any correlations with what you're seeing using sequencing data?
Yeah, we did obtain some blood samples. I would say maybe not as successful as we had hoped. We do have some markers of nasal inflammation that we were trying to look at. We have some pretty good viral respiratory data that we can try and incorporate, which Christine has been working on in the STOP2, similar to the STOP2, yeah. And then we also have some measures of smoke exposure that Ben Kopp collected for us in many of these infants.
And maybe if I can add another question, do you think that these observations are going to hold across a longer period of time? Like if you're looking at long term?
Well, I think that's probably the big question, and is a key question that families want to know. Is if we overuse antibiotics, what's going to happen, and resistance and other effects are things that they're worried about. And so I think part of the motivation for this study is where do things go from the infants who just already have this dysbiosis to the adults who have had multiple courses of antibiotics over time and now have a further dysbiosis, and where does this fit into it? So this is sort of just one step in that direction to try and determine where that switch is if it is occurring.
I have one question. Is there an association, especially in pediatrics, with intrinsic factors like exocrine insufficiency or CF liver disease with subsequent gut dysbiosis? Yeah,
I think that we really need to talk to Luke Hoffman about this, because I think that's really a central concern of theirs, is that the pancreatic insufficiency and fecal fat content is really driving a lot of this. And so I think that's perhaps why we're seeing the dysbiosis even in the absence of antibiotic exposure in these infants. And so I think the question is, in the face of that, what are antibiotics doing within it, and are they modifying it further? Yeah, so I think that was sort of the first question that had to be addressed, and then we could start to look at antibiotics on top of that.
So let's please hear it for our speaker.
Okay, next up is Kira Tomlinson, who's going to teach us all about airway metabolites.
Hello, everyone. Thank you so much for inviting me to speak. This is going to be a little bit different from what we've been talking about so far. Our lab, I'm a graduate student in Alice Prince's lab, and our lab is really interested in understanding how metabolism influences the host pathogen interactions, specifically during Staph and Pseudomonas lung infections, and how this is relevant to CF. So I have, no disclosures. And just as an overview today, I'm first going to review what the immunometabolic response is to these major CF pathogens and how it promotes their adaptation to the CF airway. And then I'm actually going to show you some new data demonstrating how this metabolic response influences bacterial persistence from the point of view of the host immune response. So just as a quick overview, we know that in CF, one of the major factors that happens in the dysregulated immune response is we get dysregulated immunometabolism, and that is to say we get the accumulation of certain metabolites that can regulate inflammation. For example, during staph infection, so this is just data from a mouse, when you infect mice with a common strain of MRSA, LAC, you get accumulation of this metabolite called itaconate, which is largely known to be an anti-inflammatory metabolite. And this response to staph is different from the response to Pseudomonas, shown here with the strain PA01, where you get the accumulation of multiple different types of metabolites, including both itaconate, as well as more pro-inflammatory metabolites like succinate. And just to show that this really is relevant to CF, we have two different sets of data. The first is showing that when you infect mice also with clinical strains taken from patients with chronic staphylococcal pneumonias, so that's our strains A1, A5, and A6, you also get accumulation of itaconate in the airway fluid of mice. And more importantly, through a collaboration with Clemente Brito's lab, we found that if you monitor itaconate levels in sputum compared to healthy subjects, patients with cystic fibrosis who are chronically colonized by different strains of Staph aureus also have accumulation of itaconate in their airway fluid. And the reason that this metabolic response is important is that it can actually directly drive bacterial adaptation to the lung. So we already have data with Pseudomonas demonstrating that the metabolic response can promote pseudomonal biofilm formation, and we actually see something similar with staph as well. We found that itaconate can inhibit staph metabolism at critical steps, namely glycolysis and the breakdown of sugars, which prevents carbon flow through downstream pathways involved in energy generation and bacterial proliferation, but actually promotes pathways upstream of this inhibition that can directly lead to the generation of extracellular polysaccharides that are used in biofilm. And again, this is highly relevant to staph adaptation to the CF airway. Through collaboration with Barbara Call's group, we found that in a longitudinal set of clinical isolates taken from the same patient over the span of 14 years, we observed that there was selection for increased bacterial biofilm formation with chronicity, and that this biofilm formation was also increased in the presence of this major metabolite. So the follow-up question we had to these studies is, how does itaconate actually impact the host immune response? And the reason that we had this question is because we know that these chronic lung infections that we observe in CF are not just the result of bacterial adaptation and persistence phenotypes, but also of this kind of frustrated inflammatory process where we no longer get effective bacterial clearance. And we know that this process is largely driven by neutrophils. So as many of you already know, there's this cycle of frustrated inflammation in CF where neutrophils are unable to effectively kill bacteria, and this leads to a cycle of frustrated inflammation where you don't get bacterial clearance, you get enhanced neutrophil recruitment to the airway and release of factors that contribute to lung damage. And so we really wanted to understand how this metabolic response that we observed promoted bacterial adaptation impacts neutrophil function. And just to give you a bit more background on itaconate and its role in the immune response, it's largely been described in the setting of macrophage-driven inflammation to be very anti-inflammatory, and this is through a wide variety of mechanisms that are summarized in this figure. As you can see, there's a lot going on here, but just to kind of break it down into two major categories, itaconate can suppress inflammation either by going back and inhibiting major metabolic enzymes involved in glycolysis or the generation of other immune metabolites like succinate, or by activating major inflammatory signaling pathways or inhibiting others, including ATF3, NRF2, NLRP3, and JAK-STAT signaling. So itaconate has many mechanisms of action, but as I said, most of them have been defined in macrophages, and we wanted to understand how it's impacting neutrophil function in the setting of CF. The first thing we wanted to do, though, was to understand how neutrophils are actually interacting with this metabolic response and whether or not they're directly responsible for driving the production of metabolites like itaconate. So what we did was we took advantage of a single-cell RNA-seq data set. So this is from a mouse that has been infected with a common strain of MRSA, USA 300 LAC. And when we looked at the expression of the enzyme that synthesizes itaconate across all these different lung cell populations, we were surprised to find that instead of seeing IRG1 expression in our macrophage and monocyte populations, we largely saw IRG1 expression in neutrophils. And this response, again, differed between staph and Pseudomonas. As you can see during our staph infection, shown in gray, IRG1 expression was largely limited to the neutrophil populations, whereas during our Pseudomonas infection, shown in black, we also were able to see IRG1 expression in the macrophage populations, suggesting that staph really induces kind of a distinct immunometabolic response from this other major CF pathogen. And as I said, this was pretty surprising to us. Neutrophils had not been previously described as a source of this metabolite, and they're a major player in lung inflammation. So we wanted to really make sure that this transcriptomic data really corresponded to a real metabolic response. And we hypothesized that then if we were to deplete neutrophils from the mouse, we would completely lose the ability to generate itaconate, specifically in the setting of staph infection. So that's what we did. We used an anti-Ly6G antibody to durably deplete neutrophils from the mouse lung and airway during infection. And you can see in both infected and uninfected mice, this antibody caused significant depletion, which was associated, of course, with a significant increase in the bacterial burden in the airway, which is expected given the importance of neutrophils in bacterial clearance. And interestingly, despite this increased bacterial burden, when we depleted neutrophils, we completely lost the ability of the airway to generate itaconate, confirming our single-cell data. Now, all of this data is just telling us that neutrophils are heavily involved in this metabolic response, but I'm sure the question you all have is, what is itaconate actually doing to neutrophil function? And we had two kind of major hypotheses for what it could be doing. The first centers around two major pieces of information, which is, as I mentioned earlier, itaconate is known to inhibit glycolysis in activated macrophages. And the second is that we know that neutrophils are heavily dependent on glycolysis for their function and survival during infection. So the first thing we wanted to do was to confirm that itaconate also inhibits glycolysis in neutrophils. So we took neutrophils in vitro and used extracellular acidification rate in the presence of glucose as a proxy for glycolytic activity. And as you can see, compared to uninfected neutrophils treated with just saline, the neutrophils that were infected with staph showed this huge increase in glycolytic activity that was significantly inhibited by itaconate, confirming that it has the same mechanism of action as it does in macrophages. But as I said, we expected that this would go one step further and actually impair neutrophil survival. So we also monitored neutrophil cell death in this assay and found that infection, of course, stimulated neutrophil cell death, and that when we added itaconate back to the media, we saw a dose-dependent increase in this neutrophil cell death, confirming one of our hypotheses. The second hypothesis we had around the regulation of neutrophil function actually centered on the oxidative burst. And the oxidative burst is a really critical process for neutrophil bacterial killing, especially for species like staph and Pseudomonas, and it's largely driven by this enzyme called NADPH oxidase. Recent proteomic studies that have identified different components of myeloid cells that can be covalently modified by itaconate have demonstrated that multiple subunits of the NADPH complex can actually be covalently modified. And so we wanted to see whether or not this itaconation actually corresponded to functional change. So the first thing we did was we monitored neutrophil oxygen consumption as a proxy for oxidative burst and found again that infection-induced oxidative burst in these cells that was significantly inhibited by itaconate. To ensure that this oxygen consumption was actually being used for the oxidative burst and not for other things like metabolic processes, we specifically quantified superoxide production, which is the major player that's produced by NADPH oxidase, and found that with infection, we got an increase in superoxide production, and this also was inhibited by the presence of itaconate in the media. And we're fairly sure that this superoxide is coming from NADPH oxidase activity itself, because when we added the molecule DPI, which is an NADPH oxidase inhibitor, to infected cells, we lost a lot of the superoxide generation that we observed. So all of that is to say that neutrophils are producing this metabolite that impairs their survival and also suppresses the oxidative burst. And we would expect that this might then play a major role in the ability of neutrophils to kill the bacteria and perform bacterial clearance during these lung infections. So the first thing we did was monitor bacterial killing in vitro. And you can see under normal media conditions, neutrophils are able to kill about 50% of bacteria in this model. But when you add itaconate to the media in a physiologic dose, you get significant inhibition of this neutrophil killing. And this corresponds nicely to in vivo data we have with mice, demonstrating that compared to IRG1 null mice that are unable to produce this metabolite, wild-type mice show a defect in bacterial clearance in both the airway and the lung. So to bring this all back around to CFTR dysfunction, as I mentioned earlier, we know that patients with CF who are chronically colonized by a Staph aureus have heightened levels of itaconate in their airway fluid, and we know that itaconate impairs neutrophil function, which is a hallmark of CF. So we wanted to understand whether CFTR deficiency itself can help promote this itaconate production. So what we did was we used an HL60 cell model, which are a good proxy for primary neutrophils that we can manipulate to introduce things like the delta F508 mutation. And we monitored the expression of the enzyme that produces itaconate in these cells during infection with staph. And we found that the delta F508 cells showed significantly increased IRG1 expression during these infections. Now, this is very preliminary data. Ongoing studies in our lab are trying to better understand this metabolic deficit and also whether or not it can be corrected by novel therapies like Trikafta. So just as a quick overview of what I've told you today, we started by reviewing the immunometabolic response to staph pneumonia, demonstrating that it's really dominated by this anti-inflammatory metabolite called itaconate, which can directly promote staphylococcal biofilm production in CF isolates. And then we also demonstrated that there's a second component to its contribution to bacterial persistence that is really focused around neutrophil function, because neutrophils are the main source of this metabolite, and itaconate can actually directly impair neutrophil killing and seems to be increased in the setting of CFTR deficiency. So there's many, many wonderful people who have helped with this project, including, of course, all the members of the Prince Lab, as well as Sebastian Riquelme, who now has his own lab and is looking for postdocs. So if anyone needs a job, definitely reach out to him. And of course, all of our wonderful collaborators in this community. And I'm happy to take any questions.
Thank you for a very elegant talk, and you are also very popular, so many questions are rolling in. So first question with the most votes, have you investigated whether hyperglycemia or CF-related diabetes influences itaconate release and subsequently staph or Pseudomonas metabolism or infection?
We have not, but that is a great question, especially because neutrophils are also highly dysregulated in other disorders like diabetes that are centered around kind of glucose dysregulation. But yeah, that's a great idea and definitely something we should look into. I'm sorry, I don't have any data to talk about.
That's your next year talk. So.
Yeah.
Next question, have you quantified other immune cell populations in the neutrophil depletion experiments? Are monocyte or macrophage numbers altered?
Great question. So alveolar macrophage numbers are the same. Monocytes are actually increased, which we would kind of expect since we have such a high bacterial burden. You would probably get increased recruitment of monocytes to the airway. But even though we have these increased monocyte numbers, we still are unable to produce itaconate without the neutrophils.
Next question, did you run an MIC on itaconate alone against staph in the absence of neutrophils? Is it directly static or cytal?
We have not done an MIC. We have done growth curves, and we know that staph's growth is inhibited by itaconate, which is really interesting because, for example, with the bacterial clearance data in vivo, you would expect that if itaconate was just impacting the bacteria, you would see the opposite effect that we're seeing. You would see increased bacteria in the absence of itaconate, but we're seeing the opposite, which is why we think it's a defect with bacterial clearance itself.
Makes sense. Okay. Next question, is itaconate detectable in CF sputum? If so, are levels changed if the new occurrence of new pathogens or exacerbations or modulators? And there's a reference / shout out to Dr. Pradeep Singh's talk.
Yes, great question. So it definitely is detectable in CF sputum. We have data in our lab, which is a collaboration with Clemente Brito, which showed that you can detect it. And at least in the data that we have, we've focused mostly on patients that are infected with either Pseudomonas or staph or both. And we found that at least in the setting of staph infection, it doesn't necessarily depend on co-infecting organisms unless it's something like Pseudomonas, which we know is a potent itaconate inducer. We haven't, at least in our lab yet, looked at the role of, we don't have any solid data yet on whether or not therapies like Trikafta or other kind of immunomodulatory therapies alter these sputum itaconate levels.
Okay, great. And last question. Great talk. Could you use itaconate supplementation to model chronic Pseudomonas or staph infections in mice?
That's a great question. We have not tried. I think one issue that we run into definitely with our staph model is that the bacteria are just so quickly cleared. And you can see even in the absence of itaconate, in the IRG1 null mice, we do see a difference in bacterial clearance, but if you look at kind of the overall time course of the infection out to 72 hours, both mice completely clear the bacteria. So it's been hard for us to kind of establish a long-term model, but we haven't actually tried itaconate supplementation yet, so it's definitely something that we can look into.
Excellent. Well, thank you so much for the talk. Oh, sorry. You're too popular.
Yeah, I'm sorry. I have one question. Really fantastic talk. So have you noticed any differences in morphology of staph treated with itaconate?
Great question. That was, all of these are great questions. That was something that I was really interested in understanding back when we were looking a lot into how itaconate impacts staph metabolism, especially because we saw some changes in the bacterial citric acid cycle, which we know can be associated with things like small colony variant formation and things like that. We haven't actually looked further into that. I haven't necessarily like plated staph on a plate plus or minus itaconate, so I can't actually speak to morphology, but yeah.
Or have you noticed any changes in, I don't know if you've ever whacked those colonies with frontline antibiotics such as vancomycin to see what happens, because you're seeing growth arrest, right?
Yes, yeah. We haven't, but that's also a great idea. Yeah, and something that we're definitely interested in trying to understand whether or not the effect is really just dependent on biofilm formation or if we're actually starting to see antibiotic tolerance because we have this metabolic deficit. Yeah.
Well, thanks.
Thanks.
All right. Thank you.
And now the last speaker for this session is Dr. Anna Zemke from the University of Pittsburgh. And I'm going to be loading your slides. And then you can use the laser pointer if you want. Yeah. Yeah, we can just click this if you want.
So I'm Anna Zemke and from Pittsburgh, and I'd like to thank the organizers for inviting me to present data from a study that we've done with UNC, looking at the impact of elexacaftor, tezacaftor, ivacaftor, which is probably the last time I'm going to say all of that, on the sinonasal microbiome. I have no disclosures. So I'm going to start out with a brief reminder about chronic rhinosinusitis and how it impacts health in CF. Very common. 95% of people with CF have radiographic evidence of sinus disease on a CT scan. Only about half of people have symptoms, and these symptoms are things like facial pain, headaches, nasal discharge, and loss of olfaction. And there's a treatment burden associated with it. Prior to Trikafta, after Trikafta remains to be seen a little bit, there is a need for repeated surgeries, medical appointments, medications, sinus washes that add to the treatment burden for people that already have a fair treatment burden. It's also important because there's crosstalk between your sinonasal cavity and your lungs. The sinonasal cavities can be a reservoir for infectious material that drains down into the lungs, has been nicely demonstrated in some publications recently. It can drain down into the lungs after transplant. Sinonasal and pulmonary exacerbations are linked in time. And the pathophysiology of this is probably pretty similar to what's going on down in the lung, where you've got thick secretions that block sinus drainage. These ostia are tiny and easily clogged. And you have ongoing cycles of infection and inflammation that ensue. So what we know about ETI and CFCRS, it's from the last year and a half. It improves symptoms within a week. 4 studies have shown this. It improves radiographic disease findings. 3 studies have shown this, 2 with CT scans, 1 with MRI. Improves endoscopic findings and leads to resolution of polyps without surgery. So basically the polyps go from looking kind of like big plump grapes down to these little shrunken little bumps. And we came into this with the study question of testing the predictions that Trikafta would change the sinonasal microbiome, specifically by increasing alpha diversity, that we'd be able to detect a change in the community structure, and that pathogen abundance would decrease with ETI. I'm also going to be presenting some baseline analysis in our cohort towards the end of the talk. So the study design, this is paired with one of the clinical studies looking at the effect of Trikafta on sinonasal symptoms. We had pre-post design, 2 visits, median and 9 months apart, supposed to be 6 months, but it happened in the middle of COVID, so everything got stretched out a little bit, and our sample size is smaller than we'd hoped due to loss of enrollment during COVID. First visit, people had an endoscopy with a swab that was collected endoscopically from the right middle meatus. They also had pretty detailed sinonasal phenotyping, including an olfaction test, a CT scan, symptom scales, sweat chloride and BMI, and spirometry measurements. We took these samples and did 16S amplicon sequencing. We used V4 sequencing. Argonne Labs did our sequencing, and then the analysis is mostly through the CHIME-2 pipeline with a sampling depth of 2,200, though you'll see figures in some other analysis that came from vegan probably here. With 30 people at baseline, and that left us with 23 completed pairs. We lost a couple people to follow up. The cohort had a median age of 27 years. A third of these individuals were teenagers, mostly down in the 12 to 14-year range. 75% had prior sinus surgery, and it included several people that were essentially asymptomatic, meaning they scored within the normal reference range for our SNOT-22 symptom scale. And this is a little bit different than some of the other published cohorts because we were recruiting through general pulmonary clinic for the most part at both sites, rather than selecting for people that already had severe sinonasal disease and were following closely with an otolaryngologist at a tertiary care center. So what did we find? Well, we found a lot of staph. At baseline, this is a taxa plot where each bar is an individual participant, and yellow is staph. We had a lower number than I was expecting of people who had microbiomes dominated by Pseudomonas. Pseudomonas is green. We had 3 folks who had a significant amount of Pseudomonas. And then we had a smattering of other taxa. So we had one person with a lot of Escherichia. We had one person with a lot of Prevotella. And you could kind of go from there. So ETI, we didn't see a change in alpha or beta diversity. No increase in face diversity, richness, or Shannon diversity within this cohort over this time period. And the populations were indistinguishable by Bray-Curtis index or unifrac or other. We tried several, and all of this was statistically not significant. So far striking out. The next question is, how did we try to define healthy here? Because that's a pretty nebulous term and maybe not a testable hypothesis as stated. So what we did was we compared our CF data set to a control set that was published in the last couple years that used very similar sample acquisition and very similar sequencing. There's possibly co-authors from the study in this room. A cohort with 129 people who had no sinonasal symptoms and at least 2,200 reads. The cohort, if you look at the paper, is actually quite a bit bigger than that, but I just wanted to look at the controls. Was able to download these data and directly compare it using our CHIME-2 pipeline and found a couple things. First of all, not surprising, our alpha diversity in this cohort is lower than that of the controls, both in terms of the number of observed taxa or richness and our Shannon diversity. It is a bit higher in our cohort than the previous published CF data sets, probably reflecting an overall younger group of people with milder disease. And we'd hope to be able to come in and say, well, our pathogens drop. But there's a couple problems with this in the sinuses right now, and this may just be perseverating. But the first issue is there's clear associations with disease outcomes and Staphylococcus in the lung and CF. But in the sinuses, we don't have the disease outcomes to really correlate with it at this point. And 1/3 of healthy individuals without CF and no sinonasal symptoms have a sinus microbiome that's dominated by Staphylococcus, which makes it hard to say Staphylococcus is then a pathogen. And I also don't have species level information here. So I don't know how much of this is aureus versus various coag-negative staph species. So what we did was there's a core sinonasal microbiome defined by these 5 genera, Staphylococcus, Corynebacterium, Moraxella, Haemophilus, and Strep, that have a prevalence of 100% across the control population and a median relative abundance of like 60 to 80%. And when you looked at our taxa plots at the beginning, I should have put a slide in with this. We've got a bunch of people who have sinonasal microbiomes dominated by stuff you just don't see in the healthy reference set, which may or may not be on the list of CF pathogen bugs like Burkholderia chromobacter. And so if you group all of these together into rarely dominant flora, those drop. If we called those pathogens, we could say we have a drop in pathogen of relative abundance with ETI. But we were limited in our ability to look at any specific taxa here due to our sample size. 2 side notes, and this is back on our baseline data set. So our cohort was younger and had less severe CRS than previously published cohorts. We did an exploratory analysis to determine if these following covariates predicted alpha diversity or were associated with different beta diversities. And this is a mix of features that predict disease severity in CF generally, like age, sex, lung function, genotype, and sinonasal outcomes like allergic rhinitis, symptoms, endoscopy scores. And we found 2 things. The first is that individuals with prior sinus surgery had lower alpha diversity. Talking to the surgeons, their first response was, well, the people who needed surgery had more severe disease, which makes sense. So maybe that would explain why they have less microbial diversity. Alternatively, these surgeries really disrupt the 3D architecture in your sinonasal cavity, and then many of the folks that had had surgeries are now on sinus washes, which periodically redistribute everything, depending on how vigorous the wash is. So I don't know whether this is causal or not, or which direction this goes, and both things could be true at once, that more disease has lower diversity, and surgery is a surrogate for more severe disease, and also that having had surgery is disruptive. So we saw that. And the second thing we saw was that allergic rhinitis affected population structure. So on the right side of the screen, you can see a PCOA plot using Bray-Curtis index for people with and without allergic rhinitis, and this was statistically significant using an Adonis test. And looking at the individual taxa, we saw 2 that differed between the 2 groups at very different relative abundances. The first at very low relative abundance is Ralstonia, and the second at the very high relative abundance we checked on a couple times is staph. Now, a limitation of this data set is that describing or diagnosing allergic rhinitis and CF can be difficult because a lot of the symptoms overlap with CF-CRS. In case anybody asks later, there wasn't a relationship between who had allergic rhinitis and who was on nasal steroids. Those seem to be completely independent of each other. And so while we observe this, I think this remains to be validated in a larger study where we've got better case definition for allergic rhinitis rather than a physician chart diagnosis. So in summary, the baseline sinonasal microbiome in our cohort is low diversity and overwhelmingly dominated by staph. ETI treatment did not change alpha diversity indices. ETI may decrease relative abundance of rarely dominant flora, also known as, we hope, pathogens. At baseline, prior sinus surgery associates with lower alpha diversity. And finally, allergic rhinitis diagnosis may affect baseline microbial community structures. I'd like to thank everyone who participated in this study, the research teams both at Pitt and UNC, and the individuals and families with CF who participated in these study visits despite being in the middle of migrating COVID lockdowns. I'm happy to take any questions.
All right, thanks for that great talk. So we have a few questions in the chat. So the first one is, have you reanalyzed your data excluding patients without symptomatic clinical disease to see if it affects your significance measurement?
So I didn't use a threshold binning with the MCID as a threshold for the SNOT-22. Instead, I used the symptoms as a variable, not as a discrete variable, as a continuous variable, and didn't see any relationship between symptoms and any of this. If you exclude the people without symptomatic disease, it was very few at this point. So I haven't excluded them, I've just used symptoms as continuous variable.
Another question that we have is, why do you think there's a significant improvement in lung health in changes in the lung microbiome, but not in the sinuses?
So I think it's premature to conclude that there's not a change in the sinonasal microbiome from this data set. If you look at the lung data sets that are showing changes, they're larger. I think if we had a data set that was about 5 times the size, we'd be able to see more and having to do less binning. And then if you looked at the talks this morning, some of the folks doing this sputum analyses are also binning pathogens as well. So it depends on how firmly you believe in the binning. So I think it's premature to say that there's not going to be a difference.
One other question that we have is, did you also measure total bacterial loads and changes?
Yeah, there was no change in total 16S burden with treatment. I don't have any species-specific data, just total 16S PCR. Doesn't change.
I have a question.
Yep.
It's more global about the sinus microbiome. We know with lung, microbiome changes are very unique to the individual. Mm-hmm. Do we know if that's the case with sinus longitudinally as well?
Yeah, I would refer the group to a former trainee of Jen Boddenberg or Catherine Arbrister has a nice paper out looking at longitudinal changes in the sinus microbiome. And so it looks like, kind of like microbiome in many spots, it's very individualized. Interestingly, she saw a bunch of individual microbiomes that swapped between staph and Pseudomonas back and forth over time. I didn't see that. I just saw staph with a little Pseudomonas before and then staph and staph afterwards. I didn't see anybody reverting backwards. So yeah, it's very individualized.
So I have a question. Since you see subtle changes post-ETI treatment in community structure, have you tried to look at functional analyses or metabolic analyses to see if it changes something that you would not capture using transcriptomic or
no, we haven't done that. We just have the 16S data. I think that the low diversity and the just sheer amount of staph is going to limit our ability to detect a lot of that. It's a good idea, and we haven't done it yet.
And maybe one last.
Sure.
I don't know if there are any studies out there yet showing that if there's a delay in colonization of the sinuses post-ETI.
Not yet.
Not yet. Okay.
I have a pitch for anybody with potential participants. We've got an extension of this study down in the 6 to 10-year-old or 6 to 12-year-old range, which at this point is largely 6-year-olds eligible for starting therapy. So if you know any 6-year-olds eligible to start therapy and their parents that live close to Pitt, UNC, and soon to be UCLA, we'd be happy to hear from you. So I don't know if there's going to be delays in colonization. You sure hope so. You really hope that in 15 years, when we're looking at these 12-year-olds that have been on Trikafta since, or, adults that have been on Trikafta since they were 12, that their sinonasal disease just looks totally different and is maybe not clinically significant, but we'll have to wait and see. That's I think, the hope for many people in the room.
Thank you. All right, so thanks very much to our speakers. Those were amazing talks, and thanks very much for your questions. And I think we've given you the gift of time. So we're all done, and you're more than welcome to leave. Thank you.